Bismillah Rahman Rahim. Very important paper one, which is just the recent one, which was taken in the March 23 exam. And of course, this I want you to look at this graph also. I'll be showing it to you later as well. And I want you to pause the video and have a look at it. There have been a number of very good questions on this graph. Now, starting with question number one, the photomicrograph shows cells from a human blood smear, and there's a neutrophil, and there's a line on it which is PQ. Which calculation, or this line PQ, we need to measure this. So just assume, let's say it is 20 millimeter. So which calculation shows the correct method to calculate the actual diameter of the neutrophil shown in the photomicrograph in micrometers? So in order to convert anything into micrometers, we have to multiply it by 1000, because 1 millimeter is equal to 1000 micrometers. So this would be 20,000. But then this would have to be divided by whatever the magnification. If something was written here like x40, so then you would divide it by the 40. So that is why the answer is A. The length of the line PQ in millimeter into 1000 and divided by the magnification of the photomicrograph. Question number two, very difficult question. I mean, it took me quite a lot of time to figure it out as well, so I'm sure it's going to be easy for you maybe. A student calibrated an eyepiece graticule using a stage micrometer. Each division of the stage micrometer was 0.01 millimeter. Now with 10 magnification and 10 eyepiece graticule units match 10 divisions on the stage micrometer. The same microscope was used with a 40 magnification, x40. So it was x10, then it was x40 instead of a x10 magnification objective lens. So that means it's 4 times more. Remember, from 10 it went to 40, so it's 4 times more. The same microscope will use the x40 instead of x10 objective lens to measure the diameter of an alveolus. The diameter of an alveolus was found to be 96 eyepiece units. In this situation, when the 10 was the objective lens, 10 is equal to 10. So now 96 into 0.01. I'm using this one. Please remember that. I'm using this one. 96 into 0.01. Now this is in millimeter. So the answer is 0.96 millimeter, and then I'm multiplying it by 1000 to get micrometers. And then I have to divide 960 by 4 because the magnification has now been 4 times. 10 fours are 40. So this is equal to 240 micrometer. So the answer is C. So this was a very difficult question. Please read the question again, and please see that how we've been able to calculate this 240 micrometer. Question 3. Which statements are correct for chloroplast and also for mitochondria? They contain 80S ribosomes. Wrong. They contain 70S ribosomes. They can transcribe their circular DNA. Yes. You see, what you have to understand is that the mitochondria have circular DNA, and they also have 70S ribosomes. So if they have 70S ribosomes, then transcription can take place. mRNA is made, and the mRNA will go attached to the ribosome, and a protein will be made. So they can transcribe this. They can translate mRNA, yes, because the ribosome is present where translation is going to take place, and they are enclosed by double membranes. So yes, 2, 3, and 4. So answer was C. 2, 3, and 4 is correct. So the answer is C for question number 3. Now coming to question number 4. Question number 4. The electron micrograph shows some cells from a root. Which cell structure is not usually found in cells from a root? Now what is A? Let's look at A. Now this is you can see this is the nucleus. So this is the nucleolus. Then D is the nuclear membrane. B is a chloroplast. and C is the cell wall. So the answer was, of course, you all know that a cell wall is also present, nucleus is present, which is usually not present in a root cell. So the answer is B. Chloroplasts are not found inside root cells. Question 5. Dialysis tubing is an artificially partially permeable membrane with pore size of approximately 22.5 nanometer. Glucose molecules have a diameter of about 1.5 nanometers and can pass through the pores of the membrane. What else can pass through the pores? No, bacteria cannot, hemoglobin cannot, ribosomes cannot, they are 25 nanometers. Yes, fructose can. So the answer is 4 only, which is D. Then coming to question number 6, what is present in all viruses? What is present in all viruses? Now, viruses are of two types. Either they have DNA or they have RNA. If they have DNA, it's ATGC. If there's RNA, it's AUGC. Now, if it's a DNA, you see, our ribose would only be in those viruses which have RNA. Now, it didn't say that. It says what is present in all viruses. Deoxyribose would only be in those viruses which have DNA. But adenine would be present in both. You see, adenine is present here and adenine is present here. Thymine would only be present here in the DNA virus. So there are two types of viruses. And if you've done A2, well, you know, of course, they're single-stranded and double-stranded. And there's single-stranded DNA and double-stranded DNA, and single-stranded RNA and double-stranded RNA, which we've never seen in any other organism. Question number 7, to estimate the concentration of glucose in an unknown solution, equal volumes of a range of known concentrations of glucose. What does this mean? It means you took 1% glucose, you took 2% glucose, you took 3% glucose, or maybe you took 1%, 1.5%, 2%, 2.5%. So you took different concentrations, known concentrations. You knew it was a 1%, it was 1.5%. Were each mixed with the same excess volume of Benedict. So you did the whole test in a similar, all the variables were controlled. After mixing the solutions, they are placed in a thermostatically controlled water bath at 90 degrees for 3 minutes. The unknown solution was then treated. So there was an unknown solution, say we label it U. Now we didn't know whether it was 2% or 2.5 or 3, we didn't know it. And the colors of the known and the unknown were compared. So you had this color standard of these, and then you compared it with the unknown one. So what is the independent variable? What is the thing that you are changing? I always tell you I for independent. What have I changed? So that is the independent variable. So the answer is A. What you changed was the concentration of glucose. You changed it to 1%, 1.5, 2, 2.5, 3, 3.5, or even different 2, 3, 4, 5, 6, 7%. Then coming to question number 8, which statements are correct for amylose and amylopectin? Now, you have to know the structure of it. You have to know the structure of amylose and amylopectin. Amylose is 1,4, amylopectin is 1,4 and 1,6. Is branched. They are carbohydrate molecules, yes, correct. They are formed by condensation reactions, yes. When the bond forms, then the water exits. So condensation reaction, they are linear molecules, they contain 1,4. You see, the amylopectin is branched, so here this one goes wrong. But they contain 1,4 glycosidic bond. Yes, this has 1,4, but this has 1,4 and 1,6. So 1,4 is common to both of them. So that is why the answer was B, which is 1, 2, and 4. Question number 9. Which statement is a correct comparison between saturated triglyceride molecules and unsaturated triglyceride molecules of approximately the same molecular masses? Now, when you read these sort of questions, you must be very clear about it. Number one, you have to be clear about that unsaturated have double bonds. Saturated do not have any double bonds. So unsaturated triglycerides have more double bonds and fewer hydrogen atoms than saturated triglycerides. Unsaturated have more double bonds. Unsaturated triglycerides have fewer double bonds and fewer hydrogen atoms than the saturated ones. The saturated ones actually have no double bonds. This is what you've got to understand. Unsaturated triglycerides have more double bonds and more hydrogen atoms. That is another thing, there are less hydrogen atoms. So the more hydrogen atom, this and this is of course totally wrong. Now we have to narrow down to A and B. Fewer double bonds, more double bonds, and less hydrogen. Now I'm just going to show you a diagram to understand this. Now if you look at this one, now just look at these here. Now there is every carbon has a hydrogen attached to it. But look at here, this carbon only has one hydrogen attached. This carbon only has one hydrogen attached. While this carbon has two hydrogen atoms, this carbon has two hydrogen atoms, has got two hydrogens. So you've got to understand is that saturated fats would have more hydrogen or unsaturated fats would have less hydrogen. And of course, this is more, they will be double bonds. There are, in fact, saturated fats have no double bonds. The double bond is only here in the glycerol and not in the fatty, but they're talking about the whole fat molecule, triglyceride. So now that we read it again, unsaturated triglycerides have more double bonds and fewer hydrogen atoms than the saturated triglycerides. So the answer is A. Then coming to question number 10, the diagram represents a molecule from a cell surface membrane. Which description of one of the labels is correct? Fatty acid at the hydrophilic end of the molecule, hydrophobic end of the triglyceride molecule, hydrophobic end of the glycerol molecule, phosphate group at the hydrophilic end of the molecule. So you see fatty acid at the hydrophilic, no, fatty acids, you see, when these fatty acids form the hydrophobic part, this is the hydrophilic part, the phosphate heads. So that is why the answer is D. But okay, but why is A wrong? Fatty acid at the hydrophilic end. No, fatty acids make the hydrophobic part. Hydrophobic end of the triglyceride molecule. This is not the end, it is the fatty acid tails, which is the hydrophobic side of the molecule. Hydrophobic end of the glycerol molecule. No, that is the hydrophilic. The glycerol is somewhere here. Okay, so coming to question number 11. Which row correctly shows levels of protein structure that can be held together by each type of interaction? So we've got to read through this. So first of all, let's revise this. What is primary structure? In the primary structure, which bonds do we have? In the primary structure, we just have the peptide bonds, and that's a covalent bond. Please remember that. Peptide bonds and disulfide bonds are covalent bonds. So in the primary structure, we have the peptide bonds. In the secondary structure, we have the hydrogen bonds, right? In the tertiary structure, we have hydrogen, ionic, disulfide, which is a covalent bond, and the hydrophobic interactions. So it says hydrogen bonds, hydrophobic interactions, covalent bonds. So, I mean, if you look at it very carefully, the answer is C. Why? Because hydrogen bonds are present in the secondary structure and in the tertiary structure. Hydrophobic interactions are only present in the tertiary structure. So this one, and covalent bonds are the peptide bonds which are present in the primary and the disulfide bond which is in the tertiary structure. So that is why this was the answer. Please remember, if you are not studying from the new edition, this covalent bond has been added in the new syllabus, which was for the syllabus for 2022. So if you are not using the new book, you would not know these new informations. Question number 12, challenging question. Which molecules contain at least 3 double bonds? Well, I know this is the saturated triglyceride will contain it, but then why would collagen and hemoglobin also contain? So let's have a look at a diagram and then see if you figure it out how. If you look at this structure, now there are only two amino acids, and when they join together, a dipeptide molecule, there are this double bond. There is this double bond, and then there is this double bond. So in hemoglobin and collagen, will there be hundreds and hundreds of amino acids joined together? So there will be a lot of peptide bonds. So sorry, there will be a lot of double bonds. The peptide bond, you see, I'm just showing you two amino acids, and I'm telling you just in two amino acids there will be two double bonds. In hemoglobin and collagen, there will be many, many amino acids. Now, if you look at a triglyceride structure, now in a triglyceride structure, again, you can see here, this is a double bond here, there's a double bond here, there's a double bond here. So in triglyceride structures, maybe, saturated ones, of course, would have no other double bonds between the CC of the fatty acid tails, but they would be double bonds here. So in a triglyceride, there would be three double bonds at least. Now coming to question number 13, which diagram correctly shows hydrogen bonding between the two water molecules? Now, this is very important biochemistry, which you must know, is that oxygen is partially negative and hydrogen is partially positive. So this is what we can see here. Here it's oxygen has been made positive. So this is wrong. Here positive, but there's no the partial sign is not there. So partial negative and partial positive. So hydrogen. So this is the hydrogen bond. So the answer is B. Here oxygen is positive. Here also oxygen is positive. Here also. Here is negative, but the negative they haven't shown you that partial sign on it. So that is why the answer is B. Which statement describes an example of an extracellular enzyme? Amylase in saliva is an enzyme that catalyzes the breakdown of starch in the mouth. Carbonic anhydrase is an enzyme that helps in the transport of carbon dioxide in the blood. DNA polymerase is an enzyme that helps build DNA molecule from nucleotides. RNA polymerase is an enzyme that involved in the process of gene transcription. So if you look at all these, carbonic anhydrase helps the transport in the blood. So it would be the carbonic anhydrase works inside the red blood cell, forming carbonic acid and all that. Then DNA polymerase helps build DNA molecules from. So this is also DNA replication takes place inside the cell. RNA process of gene transcription again inside the cell, DNA to RNA. So the answer is A because amylase in the salivary glands have cells and they produce amylase and then that is thrown out of the cell into the duct and then it enters the mouth. So the salivary gland, you see these are the cells of the salivary gland and then there's a duct. So these cells make the saliva and this saliva enters the duct and then it enters your mouth. And the saliva contains amylase, so it's an extracellular enzyme that catalyzes, and this is going to break down the starch in your mouth whenever you eat a piece of bread or a piece of roti or rice, then that is going to digest that starch into maltose. Now, I always say that A level MCQs are very, 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 very difficult and very, very challenging. So, I mean, students usually don't take me very seriously and say, "Oh no, miss, I mean, the O level MCQ paper was so easy." There's a phenomenal difference between the O level MCQ paper and the A level MCQ paper. Now, question 15, which row is correct for enzymes that catalyze reactions using the lock and key hypothesis? Now, of course, the first thing is that effect of the enzyme on the activation energy. We know that it lowers the activation energy, so it can only be this. So this is wrong and this is wrong. But now let's read this part. Shape of the active site in comparison to the substrate changes to become complementary. Now, please, if you remember, is the lock and key hypothesis, and then there is the induced fit hypothesis. In that, we say it molds around the substrate. So the changing part would be in that situation in which we would say the induced fit hypothesis. So that is why only this is correct, complementary before, during, and after the formation of the enzyme substrate complex. That is why the answer for 15 is B, because it does lower the activation energy and it is always remains complementary. But in the induced fit hypothesis, we say it molds around the substrate. So this was the thing. If you knew that, you would be able to answer this question. Question 16. A scientist investigated the rate of breakdown of hydrogen peroxide. Four experiments were carried out using different mixtures: substrate only, substrate and enzyme, substrate enzyme competitive inhibitor, substrate enzyme non-competitive inhibitor. The results are sketched in the graph. Rate of breakdown of hydrogen peroxide. Now, this is a very important part of the enzyme chapter. If you know it, well, you will be able to answer this question very, very effectively. So, which row shows the correct lines for two of the experimental mixtures? Substrate only, 1, substrate and enzyme and competitive inhibitor, 2. So, we have to look at these lines and we have to see which was substrate only. So substrate only would be which line, and substrate plus enzyme plus competitive inhibitor. Now we've got to remember substrate only. Now whenever I'm saying that, you always think there is starch, there is an enzyme called amylase, and then the product is maltose. So if only starch is present and there is no enzyme, what is going to happen? So substrate only. So if it's going to be substrate only, it has to be 4. Why is it got to be for rate of breakdown of hydrogen peroxide? So it just breaks down by itself, might break down a little bit. So the only answers could have been 4. So 4s were here only. Then which is substrate enzyme and competitive inhibitor? So this would be the one which is reaching the mark, which is slower than the original, but then it finally reaches the mark. So that would be 2. So that is why the answer is B. 3 would be a non-competitive inhibitor. This would be non-competitive inhibitor, and 1 would be just substrate and enzyme. But they didn't ask you this. 1 would be just substrate and enzyme. 3 would be substrate enzyme and non-competitive inhibitor. 2 would be substrate enzyme and competitive inhibitor. Question 17. The Changar students can't do that. It's only the students who've been diligently studying for 2 years can do that. Which of these substances can pass directly through cell surface membranes without using a carrier protein or a channel protein? So you see, when is a channel protein needed? A channel protein is needed only when there is something which is water soluble or it is polar. So glucose is water soluble and calcium is an ion. So again, it's polar molecule. Now, what can pass through, and I give you this mnemonic when I teach you this: small uncharged molecule. Small molecule or uncharged molecule. These can pass the phospholipid bilayer and do not need channel proteins. So that is why only carbon dioxide, which is a small molecule, will be able to pass. So the answer is D. Now, if I was in your place taking this exam, I would have actually drawn this. 1 centimeter cube, the surface area to volume ratio is 6:1. When the length is increased to 2 centimeter, then it becomes 24:8, which means 3:1. So what happens to it? The ratio halves. From 6:1, it has gone down to 3:1. So the answer to 18 is B. But why is it B? This is what you've got to understand. Surface area to volume ratio. So this is a cube, 1 centimeter. It said a cube. So 6 sides, 1 into 1, 6 sides, 6. You must know how to do this. You must know how to calculate the surface area and the volume. There is a simple formula for that. I mean, even if you have to look it up, you can Google it and see it. So surface area is 6 times, so volume is 1. But in 2 centimeter, 2 2s are 4, 6 4s are 24, and then the volume is 2 into 2 into 2. So 24:8 is 3:1. So the ratio halves. Then coming to question number 19. Which events are part of the mitotic cell cycle? Then coming to question number 19. Which events are part of the mitotic cell cycle? Interphase, telophase and cytokinesis. They didn't say mitosis, they said the mitotic cell cycle. So interphase is also part of that, telophase and cytokinesis. So the answer is A, 1, 2, and 3. Because mitotic cell cycle is the whole thing. Mitosis is only prophase, metaphase, anaphase, and telophase. But the whole thing is interphase, then prophase, metaphase, anaphase, and telophase, and then cytokinesis. So that is why the mitotic cell cycle by itself is the whole thing. I know it's not a very fair question. I don't like it either. I totally agree with you. Question number 20. The estimated total number of red blood cells in the human body is 2.5 into 10 raised to the power 13. It is estimated that each day 2.5 into 10 raised to the power 11 red blood cells are removed from the circulation and are replaced by stem cells in the bone marrow. Which percentage of the total number of red blood cells is replaced each day? So 2.5 into 10 raised to the power 11 divided by 2.5 into 10 raised to the power 13 into 100, and this would get you your 1%. So question number 20, the answer is C, 1%. So you have to just divide that and you would get to know the answer. Now I will continue the rest of the questions in the next video, or maybe I can just do 21. Now it says question number 21, DNA polymerase catalyzes condensation reactions between molecules during semi-conservative replication of DNA. What is joined by DNA polymerase? Base and base, base and nucleotide, nucleotide and nucleotide, phosphate and ribose. Now the catch is always this. This is where you're going to get this big stuff. Ribose is not found in DNA. DNA is deoxyribose. So that is why the answer is C, nucleotide and nucleotide. So you are going to make a mistake in the D, I know that, phosphate and ribose, because if you look at it, it does connect the phosphate and the deoxyribose, but nucleotide to nucleotide was also the correct. The rest of it all is wrong. Base and nucleotide bases, it doesn't correct the bases. So everybody must revise the DNA polymerase and how it forms the phosphodiester bond. It forms the phosphodiester bond. Then coming to question number 22, two polynucleotide strands make up a DNA molecule. Which description is correct? Now, I often say this is actually checking how good your English is. The percentage of cytosine is 50% to that of guanine. No, that's rubbish. You see, if there's one C, there's another C, then there's a third C, there'll be three G's as well. G, G, G. The percentage of cytosine is the same as that of guanine in the whole molecule. You see, this is the whole molecule. So C and G would be the same. So that is why the answer to 22 is B. But why the others wrong? The percentage of cytosine is the same as that of guanine in each strand. No, this is one strand. So there can be many A's, many G's, many C's, many T's. They're not going to be the same. That of each strand. So this is the strand. So you have an A here, you have a G here, but the G and C are not going to be. So the percentage of cytosine is the same as that of guanine. No, that's wrong. The percentage of cytosine is the same in each strand. No, that's not the case. In this side, we have this C, then we have this C, then we have this C. We don't have any C on this strand. So the percentage of cytosine is the same in each strand of the molecule. This is again incorrect. But this is actually checking how well you have read the question. Now I finish the video here and I'll continue the rest of the paper in the next video. And best of luck, and inshallah I hope you do very well in the upcoming exam. So we've completed question 1 to question 22 in this video.